Okay, so for today, just a lecture on an introduction to multivariate statistics. We'll talk about the concept of ordination briefly. There's obviously a lot more to multivariate statistics and this process of ordination than we're able to cover in this course, but I wanted to give you at least a cross-section of it. And then I also wanted to focus on principal components analysis, PCA, non-metric multidimensional scaling, Nmds. Those were two of the analyses that students in this class are really interested in using. And then just as we did for the regression analyses and the ANOVA type analyses, I've given you, in this case, a dichotomous key instead of a flow chart, but it's basically a series of questions that you consider about your data that help you to identify the most appropriate multivariate statistical method for analyzing your data, okay? And we'll cover that at the end. But in terms of ordination, ordination is the ordering of objects that are characterized by multiple attributes. So we say that is multivariate objects. And for those of you interested in community ecology, you can think about a species by site matrix where you go out to multiple locations and you evaluate biodiversity there. Maybe you classify organisms to the genus and species level. You create this matrix. That's kind of the classic multivariate analysis in community ecology. And we're going to cover that with the Nmds today. For those of you that aren't necessarily ecologists, but maybe you're interested in environmental gradients, a PCA is a useful analysis for that. So there you can take multiple measurements of some habitat feature, for example. Maybe you've got a grid of cells, or you're making observations of attributes within each of those cells. You're measuring multiple attributes, and you want to summarize those multivariate data in a smaller number of dimensions. That's another reason for using ordination. And usually for the smaller number of dimensions, we try to do it in 2 or 3. 2 dimensions, you can put that on a biplot. That's pretty interpretable. 3 dimensions, you can do a 3-dimensional plot. But some of these analyses that we'll be going over go into much higher dimensions than that. So if you're collecting 15 environmental features at each of your observational units, there's 15 dimensions to the data that you might be interested in there. And a PCA is useful for helping you to summarize those dimensions and think about maybe redundant measurements of habitat features. And so you can actually summarize data in a much smaller number of dimensions through this ordination process, okay? And there are 2 major themes in ordination. There's what we call direct ordination, there's what we call indirect ordination, which is sometimes called indirect gradient analysis or unconstrained ordination. Here we're thinking about a single matrix, so the site by species or the site by habitat features matrix. And that's the kind of one matrix-based approach to ordination. And then there's the two or more method to ordination. We call that direct ordination, sometimes known as direct gradient analysis or constrained ordination. And usually what we're thinking about there is how much variation in community structure can we explain using the environmental variables that we measured at the same locations. So you have those 2 matrices and we're constraining one by the other. And so we're going to focus first on indirect ordination today, but for those of you that are conducting community ecology, you may be interested in direct ordination, okay? And we'll talk about the different models for each of these after we get a little bit of information about the 2 that we're going to focus on today, okay? So PCA, I know that's one that at least one person in the class is interested in, but many of you have probably encountered a PCA before. And maybe you just need to know how to interpret them. And then ultimately, I'm going to show you in this class how to fit a PCA in R. It's one of the most commonly used indirect ordination or unconstrained ordination techniques. It's typically used to reduce the dimension of a data set by creating what's importantly linear combinations of the original variables. Okay, so we're going to go through an example here in this figure where we think about data from only 2 dimensions. Of course, your data will probably have higher dimensions than that, but it's very easy to think about it in 2 dimensions and then extend that understanding into higher dimensions, okay? So for the sake of this example, let's think about measuring 6 objects, and for each of those objects, we're going to collect 2 variables. So for me, I typically think about this as a site on a stream where we're going to think about things like water depth, water velocity, substrate composition. But for some of you that do morphometrics, you might be thinking about an individual specimen, and you might be thinking about head size or ratio of the body length to body depth. These objects do not have to be physical locations. It can also be used for thinking about measurements on individual objects. Okay, so it's object 1 through 6. Those would be replicates. Okay, and we're thinking about variable 1 and variable 2. Okay. And so the first thing that we can do is just plot an XY. That's what this is showing. So here's variable 1, here's variable 2 on the x-axis and the y-axis. And then object 1 is plotted at its coordinates. So along variable 1, it had a value of 1. Along variable 2, it had a value of 5. So 1 and 5. There's object 1 there. There's object 2, object 3, 4, 5, and 6. So we're plotting these things based on the scales that we originally measured the data. We're plotting an XY plot. And then what the Pca analysis does is it fits what's called an eigenvector through the longest axis of the cloud of data. Then it fits a second eigenvector that's orthogonal, or 95 or 90 degrees to that first one. And so if we consider these 6 objects here, this cloud of data, each of these points, notice that that's not a perfect sphere. It's oblong, and it's almost never heard of to have data that are perfectly spherical. There's always going to be a little bit longer direction. And that longest axis, or the longest gradient of the data, is what this first Pca axis or eigenvector is fit to. So it goes through the cloud of data so that it goes along the longest gradient. Okay, that's an important concept for understanding how Pca works and interpreting a Pca. Okay, so between object 1 and object 6 down here, that's the longest gradient of the data. Okay, so it fits an eigenvector there, and then it goes 90 degrees and fits a second one. And notice it goes along the dimensions of the it goes from the objects down here to up here, right? So it's fitting these eigenvectors to the data. Okay. And each eigenvector has an accompanying eigenvalue. You might have heard this before. And those eigenvalues represent the amount of variance that's described by each axis. And this is based on reducing residuals, or the distance between the point and that eigenvector that's fit through there. We're trying to explain the most variation with the data on the first Pca, and then the second highest amount of variation on the second Pca. And if we had a third environmental variable, we could put in a third axis in here, and it might be orthogonal to this second one that we fit there. Okay, but each of those eigenvectors has an eigenvalue, and that value is reflective of the variance that's explained by that axis. Okay, and so the axes that the eigenvectors represent are typically called principal components, or a linear combination of the original variables. And we call that a linear combination because we're fitting a straight line through there that represents a combination of variable 1 and a combination of variable 2. Okay, so that's the PC1, and there's PC2. And when you get the output from a Pca, all it does is just rotate this so that PC1 is the x and PC2 is the y, and the data are centered on the origin of those two. So notice the relative locations of object 1, object 4, 5, and 6. Those locations are still maintained in this output from the Pca, but what we've done is rotated it just a bit so that Pca1 becomes our primary axis and Pca2 becomes the secondary axis. Okay, and this would be important, of course, if we had more than 2 dimensions of data. If we had 3, we'd have 3 Pcas. If we had 4, we'd have 4 Pcas. But you don't always have to focus on what Pca 3 and 4 are showing you. Most of the important variation or the greatest majority should be captured in the first two PCAs. Okay, so this is why we call it dimension reduction. Instead of thinking about all of the variables themselves, we're creating these linear combinations to summarize the data. Okay. Is everyone doing okay? So we take the data, we plot it in an XY plot, we fit these eigen vectors, we rotate the data so that the first eigenvector is the x-axis and there's the second one, okay? And I've put a description here of what's happening there, basically everything that I just told you. And what's important is that that second is orthogonal for the first, 90 degrees between the two, okay? And the reason that that's important is because what that means is that PCA2 should be independent of PCA1, because if it were fit or if it were fit in parallel to it, those things would be cross-correlated. But by this, just the nature of putting that in orthogonally means that that is a separate axis that you can interpret independently of axis 1. Okay. Okay, so then what we do from there, so this is a powerful way of expressing high dimensional data, because you can imagine if we had more than 2 variables, we'd have 3+ dimensions, and it's hard to think about that many dimensions at once without somehow summarizing the data. What that means though is, remember, if we had 15 variables, we'd have 15 PCAs. And we know the first one's going to capture the most variation, and the second one the second most, and so on. But you'll actually end up with 15 of those. And at some point, it transitions from pattern to what's referred to as statistical noise. And there's a lot of what's called stopping rules in thinking about which of the PCA axes should we retain for interpretation. And there was a paper by Jackson in 1993 that introduced this concept. And I'll show you in R how we use what's called a broken stick model, where we think about if the variation was evenly distributed across all of the PCAs, we can think about a uniform distribution. Then we can plot against that what's it actually explained by PC1, 2, 3, and so on. And then if what's explained is higher than what it would be if it was uniformly distributed, then we retain that. Okay, so more on that on Thursday. But typically what you will see is PC1 versus PC2. And if the data aren't described in 2, sometimes you'll see a third PCA, but most of the time it's 2 or 3 dimensions, okay? So what this shows is, so this is just the same plot that I showed you earlier with PCA1 here, but now we're showing the eigenvalue, which you can interpret as the proportion of variance that's explained by that axis. So 70% or 71% of the variation in the relationship between variable 1 and variable 2 is captured along the first PCA, okay? Or 71% of the variation among these 6 objects based on these 2 variables is represented by this first axis. And that axis might be something that you would want to use to explain maybe some of the patterns that you see among these objects that we measured, okay? And then notice PCA2 is here, and its eigenvalue is 0.29. So the remaining 30% or 29.5% is explained by PCA2, okay? And so if you were to plot, and we'll do this on Thursday, if you were to plot like a frequency histogram across PC1, 2, 3, what you should see is the variance goes way down after you get past 1 and 2, okay? And you can see it here in this example, 70%, that's more than twice of what's explained by PCA2, okay? So those eigenvalues are really important information for thinking about how are we able to explain these multiple variables across these multiple objects, okay? And then the other thing that this is showing are these arrows, and these arrows represent vectors for the direction of increasing magnitude for each of the original variables. So what we can think about, so here's variable 1 going in this direction, and here's variable 2 going in this direction. So if we use PC1 to explain the kind of major emergent patterns from these 6 objects based on the 2 variables that we measured, what we might interpret is that PC1 explains 70% of that variation. Objects with scores that are positive along PC1 have higher values for variable 1. And then objects that are distributed negatively, so on the left side, remember this vertical axis is represents 0. So these would have negative scores along PC1. These would have positive scores along PC1. And so these objects that are negative along PC1 have higher values for variable 2. Okay. And so you can imagine if there were 30 variables that we were thinking about, and all of those vectors kind of aligned, you would actually be summarizing the data in only 2 axes or 2 dimensions instead of thinking about each of those individually. Okay. So when I'm going to show you a PCA plot here, but typically what you'll see is from PCA output, you'll see usually an indication of how much was explained by the first axis and the second axis and so on. Is everyone doing okay with the concept at least of PCA? Okay. So there's that Jackson paper from '93 about stopping rules. This thing's been cited a couple thousand times. It's still something that we think about, even though the paper is older. And it's something that Yari Oksanen has implemented in the vegan package. So vegan, community, ecology, and R is the package that we're going to be using. And we can fit a PCA using the RDA function. And RDA is redundancy analysis. That is a constrained ordination where you have two matrices, one of environmental perhaps and one of community data. But if you only give it one of those axes in the RDA, or one of those matrices, I should say, in the RDA function, it'll fit a PCA. Okay, and you know probably wouldn't have known that intuitively without me telling you that, or without doing the internal help for the RDA. Okay, but I'll give you some code on how to fit a PCA, and then we're gonna on Thursday, then I'm gonna let you guys fit your own. Okay. So just here's an example from a paper that I published a few years ago with some co-authors, and here we're thinking about stream fragments in the Great Plains. Each of these points represents fragment of stream between 2 dams. And for each of those points, each of those objects, we measured 3 variables. We measured the length of the fragment, how long in kilometers is it between the upstream dam and the downstream dam. We measured discharge, or how much water moves through the system on an annual basis. And then we measured the number of days that there was no discharge, or dry days, days without flow. We call that dry. So we've got 3 variables there that we're thinking about across these 60 or so fragments of stream. Actually, I think it was closer to 40 maybe. But each of those points represents an observation. And so we've got PC1 here on the x-axis, and we've got PC2 here. And we can interpret this by thinking in the context of the fragments of stream, the points that exist there. So in this paper that's published in Ecological Monographs, what we find is that fragments with positive values along PC1 tended to have low discharge. So notice discharge is increasing in this direction. So this 23 would have really high discharge. But as you go positively along PC1, all of those fragments have low discharge. And it might be intuitive then to realize that those are the same fragments where the number of days without flow, or dry days, increases. So as we move positively along PC1, we see stream fragments that have very low discharge and tend to dry often. And then as we go negatively along PC1, we have stream fragments that have high discharge. They don't tend to dry very often, and they have very long longitudinal length. Okay. So we're not thinking about individual variables anymore. We're thinking about how those things co-vary across all of these objects for which we measured information. Okay. And then from an ecological context, what you can do is overlay, like in this case, fish species richness. And so what we find is where discharge and length are greater, but there's no drying, there's more fish. But then as you move towards increasingly fragmented drying streams that don't have very high discharge, the number of species declines there. So you can actually use PC1 here as an explanatory variable that represents a linear combination of the original variables that were measured. And you can overlay other things against PC1. So here's an example of fish within the communities within each of those fragments. What proportion of the fish species spawn benthically, meaning on the bottom, or pelagically, meaning within the water column? And what we find is that pelagic spawning species are reduced in their relative abundance as we go towards these shorter, drier fragments, and benthic species increase in their relative dominance as we go towards these shorter and drier fragments. So the PC axes, I guess my point is, can be used to kind of show a summary of the data, but they can also later be used as explanatory variables, okay? And that's one of the chief criticisms, I think, of PCA, is when you create these latent variables, sometimes they're called, or these linear combinations of the original variables, it's a little bit harder to interpret what's happening there, because you've got to think about the vectors for the original measurements and how they lay out in multivariate space. Okay, so when you go with the PCA analysis, or if you're interpreting the PCA analysis, individual variables are not what you're interpreting. You could say, well, discharge increases in this direction, but you have to also recognize that drying decreases in that direction and that length increases in that direction. It could be it's some combination of those three that's causing those points to fall as they do in multivariate space. Okay, and when I say multivariate space, what I mean is the two-dimensional space that occurs here. So another thing to consider is that these points that are on top of each other and near the origin are kind of the average condition. And then as we move away from that origin, these points represent outliers along those environmental measurements. Okay, and so you could interpret it as 13 and 22 do tend to dry more often, but they also have lower discharge and shorter lengths. Okay, does that make sense to everyone? We're no longer interpreting these individual vectors. We're thinking about this entire axis, okay, and how these individual variables lay out along that axis. Okay, that's one of the main misinterpretations of PCAs that I've read about in the literature is to create this biplot, but then focus on individual variables instead of thinking about the axis itself as the main thing that you're interpreting. Okay. So I put there's a copy of this on my webpage if you want more information, although I recognize that very few of you in this class are fish heads. But that's just one way of using a PCA and the resulting gradients that come out of those in ecological study. Okay. And then so for Thursday, we'll pull some data. And if there's anyone in this class that has data that you want to fit to PCA, please bring those on Thursday, and we can do a preliminary fit. And then I'll show you how to create graphics like this in the ggplot. Okay. Any questions about PCA? Okay. Well. Spend some time thinking about it. Again, I recognize that these things mean more to you once you're thinking about it in the context of your own data. But for any of you that have collected data that are on a continuous scale that are from multiple observational units, you can fit a PCA. Notice I said on a continuous scale. That's one of the limitations of a PCA. It doesn't do such a great job of mixing continuous and categorical variables. If you have both of those, you're not without an option. There's multiple factor analysis that allows you for combining continuous and categorical variables into an output that gives you axes just like a PCA, but they're MFA instead of PCA. So that's multiple factor analysis. And I can show you how to fit those in R if there's anyone out there or in this class that's thinking that's something that you're interested in. Okay, multiple factor analysis is there's a different package. You can't fit it in vegan, but I can share with you the package to fit a multiple factor analysis if you've got a combination of categorical and continuous variables. Cool. Okay. So more on that later. And of course, please bring questions if you've got them. Then let's talk about non-metric multidimensional scaling. And this is an analysis that you're likely to see if you've spent any time around either community ecologists or ecologists that spend a lot of time collecting multiple variables for individual objects, but those variables are all scaled the same. So like in community ecology, it's either abundance data or presence absence data. So you can create a matrix where all of the data are measured on the same scale. And there's certainly conditions where you can do that with a non-community data, but most of the time it's community data that are being ordinated. Okay. So here, rather than environmental data, we're thinking about community data, and we're going to fit an Nmds. And so what this data does is, or what this analysis does, is start with a matrix where the columns represent species and the rows represent observational units. So for us, it's sampling site where we collect fish. For people that study birds, it's typically like a point count where you're standing and making counts. Is there anyone in the class that can give another example that works with other organisms where you collect community type data?
We did this in my research a lot. So we want to know how things smell, basically plants or insects. So we collect the scent or like odor cloud from around a plant, for example, and then we run it on some kind of spectrometer. So like we use a GCMS in my lab.
Okay.
So then we can identify individual compounds.
Okay.
So like what exact compound made up the odor blend for a specific plant, and then do this over many plants to kind of know over like a general odor blend from each treatment that we have.
Excellent. That's a very different example. Thank you for sharing that. That's great. So all of the different odors are measured on the same scale.
Yeah.
And you get it from your gas chromatophore, is that what you said?
Mm-hmm.
And then so they're measured on the same scale. So there's columns with measurements of the if we want to think
abundance.
About abundance of a certain odor.
Yeah.
Your columns are different plants, or sorry, your rows are different plants.
Mm-hmm.
Okay. Very good. So certainly you can fit an Nmds to that. Sounds like your lab does. Anyone else have a different example? Hopefully the two main points are coming through, that you've got to have a matrix of data and that the measurements for each of the columns need to be on the same scale. And you can transform data to proportional data to do that. So sometimes, like in morphometrics analyses, we can take links of certain traits. So I published a paper with a botanist where he did this with flowers. Relative to the width of the leaf, we measured all of these different parameters. But because we divided those by the same denominator, they were on the same scale. We scaled them, and then we could fit an Nmds and think about some of these gradients in morphology that came from that. So it's certainly not only community data, but you could think of it as a collection of points measured on the same scale. Okay. And typically what we're thinking about is either similarity or 1 minus similarity, which then becomes dissimilarity. And you'll see those used interchangeably in the literature. Some programs like Primer, some of you maybe have used Primer. This is the Plymouth routines in marine ecology. It's kind of a program that you buy that's standalone that does a lot of the Nmds analyses. And in that program, similarity is what is used. But then in R, when we start to fit some of these, you'll notice that dissimilarity is used. And so, you know, you got to keep in mind which metric is actually being analyzed, but I would recommend that when possible, use dissimilarity or distances, because there was a paper I published where I used primer and we were calling everything similarity, and a reviewer wrote like 2 pages about why they disliked that and why Bray and Curtis, who we'll talk about in a moment, actually created a distance or dissimilarity metric. So I had to go through the manuscript and change all of my similarities. I had to subtract them from 1 to get dissimilarity. So it is important to some people who may be reviewing your work, but it's used interchangeably by others, okay? But the idea, though, is that we're thinking about, based on either abundances or presence absences, how dissimilar are 2 things. And so we call those dissimilarity indices. Sometimes they're called distance. So you'll see dissimilarity and distance used interchangeably. And they're measuring the same thing. And we're going to define it here. The most common distance metric is the Bray-Curtis distance that was created by Bray and Curtis in 1957. And here we're thinking about pairwise differences between all of the possible pairwise combinations in your data matrix, okay? So here, the Bray-Curtis distance between observation or object I and observation or object J is equal to 1 minus 2 times Cij, where Cij represents the minimum abundances of species common to both samples, so that'd be species that occur in I and J, divided by S sub I, which is the total count of specimens in sample I, and then S sub J, which is the total count of specimens in sample J, okay? So to give you an example of this, I realize some of you, this is a simple formula that you might think about all the time. Some of you, this may be entirely new. So just a quick example would be if we have sample I and sample J, we're thinking about species A, B, and C. We can determine those that are common or occur in both of them. So species A, the abundance in I is 6. For species A, the abundance in J is 10. So that's common to both of the 2 samples that we want to compare. But notice species B, 7 in I, but 0 in J. So that one does not occur in both. It's unique only to I. And then species C, it's 4 and 6 across I and J. So when we say it represents the minimum abundances, what we would do is for species that are in both, so species A, there's 6 and 10, so we would use 6, the smaller of the 2 values. In species C that's in I and J, it's 4 and 6, we'd use 4, the smaller of the 2 values, okay? That's the minimum abundance between the 2. And then SI is just basically adding all of these across sample I, so 6 plus 7 plus 4. And then S sub J is just 10 plus 0 plus 6, okay? So, so Cij is 6 plus 4, and I got that from 6 is the smaller value for species A, and 4 is the smaller value for species C. And then, so that value gives you 10. Then S sub I is just 6 plus 7 plus 4, gives you 17. S sub J is 10 plus 0 plus 6, gives you 16. So we plug those into this formula here. It's equal to 1 minus 2 divided by 10 over 17 plus 16, and through the order of operations, we get a value that is 0.39, okay? So the Bray-Curtis distance between those 2 is 0.39. And it'll range between 1, which is just complete turnover between the 2. There's no shared species between the 2. And 0, which would mean they have the exact same species and they have the exact same abundances of those species, okay? So we can think about this distance or dissimilarity on the Bray-Curtis scale between 0 and 1, okay? And that's when we're dealing with abundance data, but sometimes you use only presence absence. And so I'll show you on Thursday how we can transform this using the vegan package and the function decostand to transform that into presence absence. And what that does is if there's 10 individuals there, that gets a 1 because that species was present. If there's 6, it gets a 1 because it was present, and so on and so forth. So regardless of the abundance, as long as it's greater than 0, it's coded as a 1. And then if it is 0, that means the species was absent from the collection at sample J. It retains that 0, okay? And so sometimes if you're analyzing data and you don't have equal effort among all of your samples, in which case your abundances may be skewed because of that differential in effort, sometimes occurrence is better to use. So presence, absence, this is 0 and 1 idea. And if that's the case, you might shift from using the Bray-Curtis distance to using either Jacquard or Sorensen distance. And so here's the Jacquard, so distance sub Jacquard is equal to 1 minus, and notice this 1 minus is present in all of these. That's what creates dissimilarity. We're taking the similarity, subtracting it from 1 to get dissimilarity or distance. Here it's A, so it's 1 minus A, which is the number of species that are common to both samples. And then that's divided by species common to both, species that occur only in B, and species that occur only in C, which would be, in the context of our example here, I and J, okay? So the number of species common to both is A. B is the number of species that occur only in I. So we had 3 species present. And C is the number of species, or the species that occur only in I, there's 1, that's species B. And then the species that occur only in J, there were no species that occurred only there, okay? Does that make sense? I hope I didn't trip anyone up there. Okay, so it's the species that are common to both, those that occur only in I, those that occur only in J, okay? So what we end up with then is the distance for Jacquard is just 1 minus 2 divided by 2 plus 1 plus 0, okay? And so you get the same dissimilarity or distance, but it's now based on presence absence and thinking about species that are common in our pairwise comparisons versus those that occur in only one of the 2 items that we're comparing. Sorensen's very similar to that. The only difference is that it multiplies 2 times the common species, so it weights those species that are common a little heavier than the Jacquard does, okay? And so I'll show you how to calculate Bray-Curtis, Jacquard, and Sorensen in R on Thursday. I'll give you the code to do that. And those distances are what are used for the foundation of non-metric multidimensional scaling, okay? So once distances among all pairwise comparisons in a matrix are calculated, which you can imagine would take a long time if you have a large number of species or a large number of smells that are present or a large number of morphological features that you've measured. And then you've got a large number of observational units. There's a lot of pairwise comparisons that are happening there. And so obviously R and vegan can do this pretty quickly on a reasonable sized data set. You wouldn't want to do all of that by hand, but I wanted you to just be aware of how those distances are being calculated. So then what it does is it takes the distances among samples in a starting configuration, and it thinks about the Euclidean or straight line distance between each of those. Euclidean distance is sometimes called linear distance. And in some number of defined dimensions. And this is what the really nice thing about Nmds is so like I told you, when we fit a PCA, if we have 15 variables, we're going to have 15 PCAs. We might think about PC 1 and 2, but we're leaving 3 through 15 uninterpreted, either as statistical noise or some important variation left on the table that we didn't explain. And so these stopping rules that Jackson talks about that plague PCAs, you don't have to worry about that in an NMDS because you specify the number of dimensions that you want to show, and we typically show it in 2. And then it's going to calculate this stress value. That's what this represents. And that stress value will give you an indication of whether or not you can show all of those relationships in only 2 dimensions. If your stress value is really high, that means it's having to contort the points from their original distance to form Euclidean distances that are interpretable in that number of dimensions. And if it's a really high stress, the points are being highly contorted, you would not want to interpret that output. And so this, if we're doing two-dimensional, this two-dimensional stress value that's calculated automatically when you fit this in vegan, will give you an indication of whether or not the number of dimensions that you've selected is appropriate or not. And if the stress value is really high, the answer is to just try and fit one more dimension. Instead of showing it in 2, maybe show it in 3 and see if the stress value goes down, okay? So we won't go through in as much detail how to calculate stress. It does it automatically for you. And in vegan, we're gonna fit these NMDS analyses using Meta-MDS. That's the name of the function from vegan. And it's going to iteratively move those points around in a way to minimize stress. And so when we run through it on Thursday, when you hit run on Meta-MDS, it's going to go through all of these iterations. And for each iteration, it's going to show a stress value. And it's trying to minimize that stress value, okay? So that we can get a really nice interpretation of multivariate data in the number of dimensions that we have selected. Okay, and so in two-dimensional is almost always what you see. Sometimes you'll see a one-dimensional, sometimes three-dimensional if there's really high stress, but it seems like it's almost always two-dimensional. And that's the default in R is to fit 2 dimensions. An XY plot, just like the PCA that we looked at. And then on these stress values, there's some rules of thumbs or a rule of thumb that everyone kind of follows. If that stress value is less than 0.05, what that means is that you have an excellent representation of all of the pairwise distances between all of your observational units down in 2 dimensions. It's a really excellent representation. If it's less than 1, if it's greater than 0.05 but less than 1, it's still a great representation. If it's less than 2, that's still good. You can still interpret it. But if it's greater than 2, if your stress value is greater than 0.2, what that means is the points are being contorted so much that making some sort of conclusions about their relative locations in multivariate space is spurious because they've had to be moved quite a bit to fit it into 2 dimensions. And what you would want to do there, if your stress value is greater than 0.2, is consider fitting an NMDS with a higher number of dimensions, so maybe 3. And I've seen 3-dimensional plots. It's pretty easy to make a 3-dimensional plot. 4 dimensions, much more challenging. And typically what you'll see though is in the literature, if 3 dimensions are fit, they'll show NMDS1 versus NMDS2, and then in a second panel, NMDS1 versus NMDS3. So that's one way of showing 3-dimensional data in just a 2-panel plot, okay? So if I lost you in the distances or if I lost you in the stress calculation, let me just tell you how to interpret an NMDS plot, okay? Because that's basically what many of you might need to know if you're reading the literature and thinking about some of these things that are analyzed with NMDS. So here's 2 dimensions. Here's NMDS1, here's NMDS2. This is from a paper that some students and I published just a couple years ago. It's on fishes again. But notice we report the stress value, so 0.09. What that means is it's less than 0.1, which means it's a great representation in 2-dimensional space. And here, instead of points like in the PCA where it was just a point with a little number next to it representing it, what I'm showing you is the letter 4, that's sampling site 4, in the year 2015. Here's sampling site 4 in the year 2016. And there should be a sampling site, oh no, it was just 2 years, so we collected fish in 2 years, 2015 and 2016. And the way that you interpret this is what that means is that because 4 here in 2015 and 4 here in 2016 are very close to each other, that means they have a very small Bray-Curtis distance. We use the abundance data of all of the fish species that we caught there to calculate Bray-Curtis distance and then plot it in an NMDS. And if they're really close to each other like this, what that means is they share species. They tend to be more similar or less dissimilar. So that could mean they share species and those species have similar abundances, okay? But 4 versus site 10 over here, Those are much more dissimilar to each other because they're further apart in multivariate space. And so points close to each other tend to have comparable community compositions or similar community compositions, and those that are further away from each other tend to have greater dissimilarity, either through species turnover, which means there's different species here compared to down here, so they don't share very many species, or the abundances are much different, okay? So when you're looking at these, you can still overlay environmental variables through a process called InviFit. I'll show you how to do this on Thursday. And what it's looking at is correlations at each of those sites with things like developed land, cultivated land, forest land cover, stream order, which is a measure of stream size, and drainage area. So we can think about the communities in the background here using an NMDS, but then we can overlay environmental covariates on there and interpret that in a way that's consistent with a PCA. And so notice I'm interpreting NMDS1. I call that a stream size gradient. So here, those locations like site 10, they tend to have high drainage area, a larger stream order, a lower cultivated land cover. So larger streams. But site 3, for example, has small drainage area because that vector is going in the other direction, and it has a small stream order because that vector is going in the other direction. So we have this gradient from large streams to small streams along the first NMDS. And then along the second NMDS, we've got primarily forested, higher stream orders to urban. That's what developed land represents, to developed land. So we have a land cover gradient from forest is positive and urban that's negative there. And then you can overlay species scores. So this is panel A. I cropped out panel B, but it shows the same thing, each of the sites, but instead of vectors for environmental data, we put species scores on there. And I cut those out because I know species names don't mean anything to you guys, but certainly those data will mean something to you when they're your own data. Which of the smells, if in that example, are associated with this first NMDS axis, positive and negative? Which of the smells are along NMDS axis 2? But the really nice thing about NMDS is that you can add these environmental correlates on there, so you can still think about environmental data and community data, but it's not constrained ordination like a redundancy analysis or a canonical correspondence analysis. Okay, and I'll tell you what I'm going to explain what those are in just a minute. Okay, so the main take-home message when you're interpreting an NMDS, what's the stress value? That's the first thing you should look at, because if that stress value is very high, don't bother trying to interpret it, because what it's the visual representation in however many dimensions that you selected is not a good visual representation of the higher complexities of the data. Okay, so you want to see a stress value that's less than 0.2 before you spend time interpreting it. Again, if it's higher than that, add a third dimension on there. In this case, we had done 2. We want to add a third one. And then you can think about the points. What are their relationships? So we would expect that this sampling site 4 would be really consistent between the 2 years. And notice that site 7 is next to site 7. Site 10 is next to site 10. We would expect that the same species would occur at those sites if we went there 2 years in a row. But there's some, like site 1, for example. Here's one here. I think that's 1 there. And site 3, here's 3, and here's 3. Some of these sites that aren't really close to each other in multivariate space between the 2 years, those represent sites where the community is changing through time. And we can represent that in an NMDS just by the nature of showing the year for those 2. And there is a way of testing for differences either through time or through space. It's called a permuted multivariate analysis of variance, a per MANOVA. And I will show you how to fit a per MANOVA on Thursday. If you've got questions about, well, these, in Natalie's example, these plants all came from this field. I don't know, I'm not a plant guy, and then these plants came from this field. You might want to test whether or not the smells in those 2 fields differ from each other. A per MANOVA is a way of testing that. And an NMDS with their points colored by field is a great way of showing how those smellscapes, or whatever you call it, differ between the two locations if you were to sample two locations, right? In many ways, NMDS with environmental variables overlaid like this has replaced constrained ordination like a CCA or an RDA. There's still times in my lab where we fit redundancy analyses, where we're thinking about constraining the community data by the environmental data. I'll show you how you make those decisions on the next page. But this, I have noticed NMDS are popping up more and more. Most of the time, if there's community or high dimensional data, a really great way of showing that in just 2 dimensions without worrying about higher dimensions that have been excluded is to use an NMDS. Okay. Any questions about an NMDS? You can read this paper if you've got if you've got a high tolerance for fish information and you want to get an idea of how the data are collected and how the NMDS results are reported and what the figures might look like. Okay. We'll move on unless there's questions about that. It might seem like I have just selected PCA and NMDS out of this seemingly endless sea of multivariate statistics that are out there, and I recognize those that are new to multivariate statistics. That can be kind of worrisome because you might be thinking, what else is out there that I might want to use beyond those two? And so that's what this little key represents. And if you read that, there's a book that I cited on the syllabus about numerical ecology in R. If you download or buy that book, it covers how to do all of these analyses. And so I pulled these from an exhaustive list from a textbook. Okay, but as I told you earlier, there's direct gradient analysis or direct ordination or constrained ordination. Those are used interchangeably. They all mean the same thing. And then there's indirect gradient analysis or indirect ordination or unconstrained ordination. And that's the two that we talked about today. We talked about a PCA and a NMDS. So let's think about those for just a second. So if you're thinking about, I have a single matrix and I want to describe that, or a matrix or a table of data, and I want to describe that in a smaller number of dimensions than the raw data, you'd want to do an indirect gradient analysis. And so then you go to 6. At 6, if you have only distance values, you would go on to 7. So we created or went over 3 distance metrics today. We did Bray-Curtis distance, Jacquard, and Sorensen. There's many others that are out there, or you may have Euclidean distance or geographic distance where you only know the distances. So if you're thinking about distances, you can go on to 7. If it's what we refer to as a monotonic response, you can fit a principal coordinates analysis, or if it's what we call a non-monotonic response, you can fit a non-metric multidimensional scaling. And to give you an idea of monotonic versus non-monotonic, if you were to fit the relationship between distance, geographic distance, and community distance, and you see either a distance decay relationship or you see this increasing relationship where the slope never reverses, we call that monotonic, okay? Versus non-monotonic, where the maybe the distance starts to, or dissimilarity increases with geographic distance, but then it starts to become similar again. This might be a really long gradient. That's what we refer to as non-monotonic, and it can go the other direction. Your slope can be trending in a negative and then reverse and go back to a positive. That's typically referred to as non-monotonic. Okay. And so if you have monotonic relationships, you can do it with the principal coordinates analysis. If it's non-monotonic, you can do the non-metric multidimensional scaling. And what I see is a lot of scientists will fit the non-monotonic model even to data if they're monotonic, because these are just capable of handling a much wider variety of data. You could think of it as a parametric versus non-parametric kind of. Do we fit this monotonic assumption versus the non-monotonic assumption? If you go straight to non-monotonic, then it doesn't really matter what your data look like. you can still fit the model. So that's one of the reasons that non-metric multidimensional scaling is so often used. That's if you have only distance data. Sometimes you have the raw data, and so if they're count data, or if they're the relative abundance of smells, for example, you can go on to 8. If you have monotonic responses, you can go on to 9. There's principal components analysis on a correlation matrix, or there's principal components analysis on a covariance matrix. And we'll talk about this on Thursday when we fit these PCA models. Are we interested in knowing how the variables correlate with each other, or are we interested in thinking about how those variables explain covariance of the observations that are there? Okay, and we'll spend a little bit more time talking about that on Thursday, but there's a couple of things you can think about when you plot a PCA. Most of the time, it's correlation. We want to know how the individual variables that we measured correlate with one another so that we can reduce the dimensionality of that. But sometimes you're in much more rarely, you might be interested in covariance between those, okay? And that you can fit with monotonic responses. If it's a non-monotonic response, again, you can go back to Nmds. If you're not interested in what's referred to as species scores or individual site scores, if you do want those individual site scores, you can fit a correspondence analysis. These aren't used very often anymore, but in a correspondence analysis, sometimes your data follow what looks like a horseshoe or an arch effect. And there's some debate in the literature about whether or not that's meaningful or not. There's some ways of accounting for the arch effect. That's called detrended correspondence analysis. And then if you do think that hump or arch effect is important, you can just fit a typical correspondence analysis, okay? If you're fitting these, you have taken a deep dive into multivariate statistics, okay? Most of you won't fit these, but if there's anyone that thinks that you're interested in doing that, I can show you how. I can show you how to fit these in vegan, okay? So that's the indirect or unconstrained. The other option, and there's fewer of them, is the direct gradient analysis. So then you go to 2, and your first question is, do you have multiple species or multiple smells or multiple morphological features, or are you just doing it with a few? Because if you're just doing it with a few, you can go on to 3, and you can actually fit linear regression or generalized linear regression if you're thinking about monotonic versus non-monotonic gradients, okay? So sometimes you'll see that as low beta versus high beta, but this is what I was talking about earlier. Monotonic basically follows the assumptions of a normal distribution. Non-monotonic is when we start to think about deviations from that, okay? So if you're only dealing with 1 or 2 species, you might want to just fit a regression model, but most of the time in community ecology, we're dealing with much more than just a few species. So then we might go to 4. We want to think about the same monotonic versus non-monotonic. If it's monotonic, if it's a short gradient, a redundancy analysis will work well. And that RDA is, as I was explaining earlier, remember, for all of these direct gradient analyses, it's 2 matrices. It's and usually in community ecology, environmental or spatial data, and then community data. So RDA, because it follows a monotonic response, if you only give it 1 matrix, it does the monotonic version, which was how we got to 9, by the way. Monotonic sent us to 9. It fits a PCA, okay? So when we are running our PCAs on Thursday, we're going to use the RDA function from vegan. And then if it's non-monotonic, then you can go on to 5, and you can fit a canonical correspondence analysis. Or if you follow this debate in the literature and the arch effect is something that concerns you, you can fit a detrended canonical correspondence analysis, okay? Again, that's a deep dive into multivariate statistics. You're probably never going to fit a detrended correspondence analysis. The ones that I see most often, non-metric multidimensional scaling, principal components analysis, redundancy analysis. Those are the main analyses that I see in my little world of ecology. But if you're in this class and you do see detrended correspondence analysis, maybe you're working in a different realm than I am, and you want to know how to fit that, we can do all of these in vegan, okay? Okay, so I saved this as a PDF. I put it on Canvas, you know, print that thing off or save it on your file. Come back to this someday when you have your own data and you're ready to do some of this stuff. Or if you're reviewing a paper and you're not sure why they selected the analysis that they did, you know, you can come back to this later, I guess is my point. Any thoughts about any of this? So we'll spend Thursday, I'm going to develop code and give it to you so that you don't have to hack all of it out. But then I'm going to have a coding assignment where you either change the model or fit the model to different data. Is everyone okay with that approach? I mean, there are alternatives where you do write out all of the code yourself. I like give you a printout or something and you have to punch it in. Most students don't like to do it that way, but I'm open to you know, doing it the way that you guys find most useful. Any discussion on that? No? Okay. Fair enough. Let me stop this recording.